In order to fully grasp biological concepts and the data that supports them, one needs to get a firm, established background in the use of biological terms that may seem arbitrary, intimidating, and confusing at first, but are actually used in biology quite frequently. This is especially important for audience members that hopefully will exist soon, that do not have much of a background in the life sciences and don't have a background in being taught the life sciences, but would still like to learn further on this channel. Elaborated terminology reserved for scientific disciplines like biology is absolutely necessary in order to unambiguously describe information as it is observed in reality, thus allowing it to be understood by more people than who just saw it firsthand. One of the most important subgroups of biological terms are directional terms that allow scientists and learners to orient themselves relative to a particular specimen. There are two different ways to go about teaching anatomical terms, and I will be teaching the non-human version given the fact that I am almost never going to talk about people on this channel in a scientific manner, which means that I'm going to be using the anatomical term subset that's used for mostly vertebrate animals that are quadrupedal. In order to fully grasp these terms, it'd be best for you to visualize an animal with its long axis, the line going through its vertebral column, its spine, being parallel to the ground, kind of like a human on four legs. Keep in mind that directional terms are assigned assuming this position, even if the specimen you're looking at isn't in the anatomical position. The relativity applies to directions like left and right. It won't be your left or your right. It'll always be the left and right of the specimen. There are a set of terms used to describe a direction going towards the head of an animal that you're going to be looking at. These are cranial, rostral, or most commonly anterior. You may want to use rostral or cranial, which is fine, because those are parts on the head of an animal or the head itself and relate to the direction a lot better than maybe just saying simply anterior. I will be using anterior just because it's the most common and the most familiar to people. Terms referring to a position that is close to the rear or tail of an animal are posterior or less commonly caudal. Caudal means tail. And you may want to use that also because it correlates better with the direction, but I'll be using posterior. Dorsal refers to the position towards the back of an animal, and ventral refers to the position towards the underside of the animal or the ground. Medial refers to a position close towards the midline or the long axis of an animal, and lateral refers to a position that deviates from this and is closer to the sides of an animal. When referring to the limbs of animals or the extremities of trees, like their branches sometimes, but normally animal limbs, proximity towards the joint that links that limb to the main body of the animal, the part that contains the midline of the animal itself, is used to orient people in regards to positions on the limb. So proximal refers to parts that are of close proximity to this area. Distal refers to the opposite, which are positions that are further away from this joint that links the limb to the trunk of the animal. Keep in mind that this is only applicable to limbs of animals and occasionally is used for parts of trees like branches and leaves. And additionally, when I say main body, that's the body of the organism, the part of the organism's body that holds the midline. Some less common ones that you might hear about are superficial and deep. Superficial refers to parts close to the surface of an organism, and they're normally used with parts that envelop an organism. For example, skin and the epidermis is superficial to fat underneath your skin. Deep refers to parts closer to the inside of an organism relative to something that wraps around it. For example, the lungs are deep to the rib cage. It is also useful to think of directional terms as they are associated with imaginary planes cut through an organism. This is especially helpful when one is looking at a cross-section where someone literally cut through an organism, and you need to orient yourself and figure out what and where you are looking at on the organism. There are three main plane types, and one of them has two subtypes. So the first one would be the mid-sagittal plane. The mid-sagittal plane is the plane that cuts an organism into equal left and right sides, and it's basically going through that midline of the organism. A parasagittal plane is a plane that is parallel to the mid-sagittal plane, but it doesn't necessarily have to cut the organism into equal, perfectly equal, to be exact, left and right sides. There is also a transverse plane, which cuts an organism into the front and back, or anterior and posterior sides. and these don't need to be of equal length ever. There's also what is known as a frontal plane, which cuts an organism into dorsal and ventral parts, but that doesn't need to be of equal size either. The only one where size needs to be equal on both sides is mid-sagittal, and that's left and right. This will be my favorite part, phylogeny and systematics. So phylogeny, for those of you who don't know, is all about the patterns among and relationships between organisms in regards to their continuous evolution. When scientists make assessments of the phylogeny of certain organisms, they often refer to them as taxa, or the singular version, taxon. A taxon is any evolutionarily significant grouping of organisms, anything as exclusive as a species to as inclusive as a kingdom. Given the fact that evolution is continuous, that means it happened in the past, and that then means that there is only one true specific way that the organisms that we're familiar with evolved. Scientists conduct phylogenetic analyses as a means of forming a hopefully robust hypothesis of what the true phylogenetic tree looks like, or what a part of it looks like. They do this by making a phylogenetic tree that is also known as a cladogram. The use of a metaphorical tree to reconstruct the phylogeny of organisms is very commonplace in taxonomy and classification as a science. After all, evolution is a branching process because populations will end up branching from one another when they separate and become isolated reproductively. It's also important to note that if one went back in time to trace the trajectory of the phylogenetic tree branches backwards, they would notice that they would meet at a node where the two branches become one. We now know that pretty much the only socially contributive way that scientists can classify organisms is by doing so according to their evolutionary relationships relative to each other. To be fair, we'd be quite far behind in society if we only classified animals in 3 categories, like livestock, pets, and vermin. It seems easy at first, like a cow is livestock, a dog is a pet, and a flea is vermin. But there are some that are incredibly ambiguous, to the point where the classification scheme completely falls apart. It's not even like you can put one in the other category and have it settled there with new evidence. For example, lizards. Lizards can be livestock to some who want to make boots and belts out of them. They also might be pets for people that like them, and also vermin for people that hate them, or for people that have to deal with lizards that eat their pets or livestock. Genetic and morphological characteristics are inherited from common ancestors, and changes in them can be acted upon by natural selection, and passed on to other generations that might successfully make up more, less, or even the majority or entirety of a population than previously. This is evolution. This means that these are traits that taxonomists study to determine an organism's phylogeny. I find it instructive to think of recovering evolutionary patterns as something similar to a really challenging game of connect the dots. Some dots might connect to each other before others, maybe not, but regardless, you need to have evidence, and lots of it, to justify either choice. As stated previously, these cladograms are hypotheses, which means that they're prone to revision and falsification when new strong evidence arises. This means that in your game of connect the dots, you might need to erase some dots and redraw them elsewhere when you have evidence for it. What we are trying to reconstruct within the cladograms are what are known as clades, or monophyletic groups, which are a taxon that includes the most recent common ancestor of 2 select subtaxa and all of its descendants. What scientists certainly don't want are what are known as paraphyletic groups and polyphyletic groups. Like, what's the point of studying a group of organisms and how they interact in an ecosystem that we're almost certainly involved in when you're not even going to include some of them? Paraphyletic groups, or grades, include some descendants and a common ancestor, but not all of the descendants. And polyphyletic groups include at least some descendants, but not the most recent common ancestor. To put it simply, paraphyletic groups exclude descendants, and polyphyletic groups exclude ancestors. Before I continue, it is important for me to say that there's virtually no way of finding the actual common ancestor of any particular taxon. It's also important to say that the ancestors of these taxa are going to be populations and not pairs or individuals. In phylogenetic analyses and accounts of the phylogeny of organisms, these ancestors are always assumed by default to be hypothetical. It's important to keep in mind, though, that in terms of finding common ancestors that actually exist and aren't hypothetical at all, there are very few rare but reasonable exceptions. Regardless, the point of taxonomy now is not to find the ancestor of a group, but rather to affirm that one does exist between 2 taxa, without either taxa being more closely related to a taxon that is excluded. You may also be asking how traits link taxa together by common ancestry, and what scientists look for to answer your question are what are known as synapomorphies. But before we get into those, let's look at some other similar terms before we get to that. Plesiomorphies are traits that are ancestral, and when they're shared, they're what are known as symplesiomorphies. Neither plesiomorphies nor symplesiomorphies are very informative about evolutionary relationships, since one can't accurately link taxa together based off of a trait that was inherited in a common ancestor that isn't even included in the group. An example of this would be, let's say, someone trying to justify that humans and chimps are more closely related to each other than either is to a mouse, and your justification being body hair. This may be alarming to some people, especially if you know that all mammals alive today have hair, used to have hair, or have traces of hair. And after all, you can't separate mice from a group containing chimps and humans and have your evidence be a trait that they all have. Homoplasies are traits shared by two taxa that have no bearing on their evolutionary relationships. These are also referred to as analogous structures, and they're caused by convergent evolution when certain organisms evolve similar countermeasures to common environmental pressures. Homoplasies aren't informative about evolutionary relationships, since they only really indicate superficial similarity, and that similarity breaks apart when one looks at it more closely. There are three ways to test for structures being analogous, and therefore falsify their homology. I'm going to list them in increasing reliability, but also in increasing difficulty to perform. Firstly, homology can be falsified if the structures are fundamentally dissimilar to each other, like insect and bird wings. Bat and bird wings are homologous in terms of their musculoskeletal forelimb structure, but not in terms of their wing structure or forelimb shape for this reason. For example, bats and birds did inherit their forelimb from a common ancestor, but they didn't inherit their wings from a common ancestor that had wings too. Secondly, a phylogenetic analysis, which will use a combination of traits to determine how organisms are related, can show that two structures have independent origins. Thylacines, which are a type of marsupial, and wolves did not evolve from a common ancestor that looked like a dog because both share many more fundamental traits with many other animals that don't have a dog-like morphology. They are therefore closer relatives to those other animals, since it's more likely that they inherited that suite of features from a different ancestor than if just the dog-like features were inherited by the thylacine and wolf from a more recent common ancestor, or if all of those animals more closely related to those two other animals lost their dog-like traits. This is using a concept known as parsimony. If you need help understanding what parsimony is, you should use that previous example as your basis for your help. But finally, thirdly, if both structures that one might be suspicious of being homologous or not are present on the body of one organism at the same time, then they definitely didn't come from a common structure on a common ancestor. Angels would reject the homology of bird and human forelimbs if they actually existed in a manner that was scientifically applicable. You may like a real example. Throughout geologic time, many lizard-like reptiles glided through the skies during different time periods. 42 are alive today in the genus Draco, which glide through the forests of South and Southeast Asia on their elongated dorsal ribs with membranous skin in between the individual bones. During the Permian and Triassic period, roughly 250 million years ago, similar reptiles known as Vigeltosaurids glided in their habitats too. But is the gliding apparatus of Vigeltosaurids homologous to the gliding apparatus of Draco? They are not homologous because Vigeltosaurs glided on unique rods of bone, not ribs. And we know this for certain because Vigeltosaurids have both standard ribs along their entire vertebral column and these separate gliding bone rods that are known as patagial spars. Thus, the Dracos and Vigeltosaurids did not both inherit their gliding apparatuses from a common ancestor's ribs, let alone a common ancestor's gliding apparatus of any kind. Modified traits that distinguish one taxon from another are referred to as apomorphic, and when multiple taxa share this distinguishable trait is referred to as synapomorphic. This is exactly what evolutionary biologists are looking for, since it indicates a point where a common ancestor inherited a unique trait and then passed it on to his descendants, thus helping scientists find an actual monophyletic group. It must also be noted that this trait must also not be falsified as homologous by the previous tests listed before in order to be synapomorphic. Some more stuff about parsimony in terms of a phylogenetic analysis and the accuracy of it in determining a phylogenetic hypothesis. The more synapomorphies, the better. A combination of synapomorphies may even be its own synapomorphy for a clade, and this can then be used as supporting evidence for a clade's existence, often regarded as a proper noun in the literature, and not just any clade, but one that actually exists. The final set of terms is about taxonomic nomenclature, and taxa are named in a unique but seemingly sophisticated manner as to prevent confusion caused by common names used in the common vernacular. For example, there would be a lot more confusion about what a mountain chicken is if it weren't for its scientific name Leptodactylus phalanx, with the genus Leptodactylus meaning slender finger, which is well known by herpetologists at least to be a genus of frog. Yes, the mountain chicken is a frog, and the scientific name helps prevent people from thinking it's a bird. Binomial names are given to individual species of organisms, which come in two parts as the name implies. The first part is the genus, which is equivalent to one's surname, except that the genera have to be completely unique as to again prevent confusion. A genus, like a surname, includes many individual types of organisms that may even be reproductively isolated. It's kind of like a family name, a surname, containing many different family members that have different first names, but the exact same last name, kind of like the same genera. The second part of a binomial name is the species name, also referred to as an epithet, which kind of acts like a first name, and it gives every organism its complete, unique, scientifically applicable label. Our species name, Homo sapiens sapiens, includes the genus name Homo and the species name Sapiens. That third name at the end, the second sapiens, is a subspecies name to further distinguish taxa that are quite similar to each other and almost certainly capable of interbreeding. But they may be parts of prominent populations or domesticated breeds. The subspecies name acts like a middle name, not entirely necessary, but nonetheless adds some specificity that can be helpful in certain situations. More inclusive taxa that include genera and species themselves have been referred to as things like families and kingdoms, but I personally don't like using those. Here's why. Some may be familiar with the ranked hierarchy that includes domain, kingdom, phylum, class, order, family, genus, and species. But I really only like using the last two since a ranked hierarchical classification scheme has been shown as of recently to be quite flawed. While evolution naturally makes clades hierarchical as is expected of it, so that clades branch off to include smaller clades and are parts of larger ones themselves, it works along a continuum that is largely free from restrictions caused by at least some man-made classification schemes. For example, reptilia and aves have traditionally been referred to as part of the class rank, even though we now know that aves are a reptilian, a dinosaurian to be exact. In this case, a clade that has given rise to another clade should always have a name that is of higher ranking than the clade that it just made, but of course, reptilia and its descendant aves have the same ranking name. Additionally, it's entirely possible that in the past, and this will probably happen in the future too, species have given rise to such a large number of other species that they would probably be named as a family, and this would thus also violate the named hierarchy, a smaller rank giving rise to a much bigger rank. Finally, the realization that there are many subclades transitional in the amount of exclusivity between ranked clades has been problematic. To put it simply, arguments over whether a certain taxon is a subphylum, a phylum, a superphylum, or a subregnum are completely missing the point, and that's exactly what you should think given the fact that evolutionary relationships have been such a large part of our conversation as of recently. And although I may mull over whether a certain species is within a larger clade or just outside of it and part of a larger clade that includes it and the other smaller clade, I'm not going to be arguing with myself over what name or what ranking those taxa deserve. Because really, no taxon has anything special about it that would warrant such a system in the first place. One argument is over how recent of a common ancestor is shared between two given taxa, and the other is over nothing as remotely significant. It's literally just, what name is going to be assigned to this clade that already exists and already is just as hierarchical as it would be without the name? That is exactly why I'm going to refer to taxa that are larger than genera and would otherwise be ranked simply as clades. For example, the great ape clade instead of the great ape family. And if I do flip-flopping between family and clade or kingdom and clade, that's purely accidental and that's just out of habit. Old habits die hard. Now that all of that invaluable, useful information has finally been described to you, I certainly hope that this video provided a deep and informative guide on how to understand what I'm even saying.